So we'll have a brief introduction about carbohydrates, which are one of the three major macronutrients in the body. Carbohydrates function as the body's primary source of energy. They are organic compounds made of carbon, hydrogen, and oxygen in the ratio CH2O bracket N. Moving on, we start out by having monosaccharides, which are singular units. For example, you have glucose, fructose, and galactose. These are your basic building blocks for carbohydrates. Moving on, we have disaccharides, which are two monosaccharides linked by a single glycosidic bond. An example would be sucrose, wherein you have a combination of glucose and fructose, or lactose, where you have a combination of galactose and glucose. Next up, we have oligosaccharides, which are composed of 3 to 10 units of monosaccharides. An example is raffinose. Beyond that, we have polysaccharides, which are composed of more than 10 units of monosaccharides. Examples of these being starch, glycogen, and cellulose. When it comes to functions, which I have not put in the slide here, but I'm just going to talk about them briefly. We have polysaccharides used for storage. An example would be glycogen, which is highly branched. The benefit here being rapid glucose mobilization. And then you have other functional polysaccharides. For example, you have cellulose in plants, and then you have glycosaminoglycans, GAGs, and so on.
Okay, let me present about my part. So monosaccharides. Monosaccharides are the simplest form of carbohydrates and they consist of a single sugar unit that cannot be broken down into smaller carbohydrates. This means that monosaccharides are carbohydrates that are the simplest units that you can find anywhere. And they are composed of only carbon, hydrogen, and oxygen at the simplest ratio. It can be classified into a few different forms just for further understanding. So let's see for the first one, classification is form. They can be drawn by two ways, which is the Fisher projection and Howard projection. In the Fisher projection, it is drawn in a linear form, where you can find small amounts of this form in a glucose solution, for example, while the cyclic form is abundant and you can find it almost everywhere using the Howard projection. Next is isomerism. Isomerism is a compound that has the same molecular formula but different structural formula. For example, we see glucose right here. They are the exact same but they are different isomers, which is D-glucose and L-glucose. They are a complete mirror image of each other and it is just flipped apart. Next, we move on to types. Types is in means by functional group. So first, we have the two main functional groups, which is aldose and also ketone. So in aldose, you have the aldehyde functional group. For example, monosaccharide such as glucose and galactose have the aldehyde group, while fructose here has the ketone group. So that is how you can classify them. Next, we move on to disaccharides. Disaccharides are carbohydrates that are made out of two monosaccharide units. These two monosaccharide units are linked by glycosidic bond, and their general formula are C12H22 and O11. For example, as you can see here, fructose, lactose, and maltose are the three main disaccharides, and they are formed by multiple monosaccharides. Let's understand glycosidic bond. It is a covalent bond that links two monosaccharide units to form a disaccharide or polysaccharide, and it formed by condensation reaction, which is loss of water molecule while forming the bond. And we have two of it, which is alpha glycosidic bond and beta glycosidic bond. And you can find alpha glycosidic bond in mainly starch and glycogen, while beta glycosidic bond you can find in cellulose. Alpha glycosidic bond is just basically the joining part of the bond is below the plane of the ring, while in beta glycosidic bond it is above the ring.
Next, we have oligosaccharide, which is a short carbohydrate chain made of 2 to 10 monosaccharides. They are usually present as glycoproteins, glycolipids, and proteoglycans, which form the glycocalyx. Their function is for cell-to-cell recognition, for example, blood group determinants, protein folding and stability, as well as cell signaling and adhesion. Example include starchose and raffinose. Next, we have polysaccharide. Polysaccharide are polymers containing a large number of monosaccharide units, and they are usually serve as storage macromolecules because of their typical features where they are insoluble, so they do not disturb the osmotic balance, and they are compact, which allow them to store a lot of energy. Polysaccharide can be divided into homo and hetero. Homopolysaccharide composed of one type of monosaccharide, whereas heteropolysaccharide compose two or more types of monosaccharides. The first example is glycogen, which is a storage molecule in the animals, usually found in the liver and muscle. Monomer is alpha-D-glucose. Bonds include alpha-1,4 and alpha-1,6 and is highly branched, which allow rapid glucose release. The storage molecule in plants is called starch, usually consists of two components, amylose and amylopectin, and is less branched than glycogen. However, the monomer is still the same as that of the glycogen. Cellulose in plants also being considered as polysaccharides made of beta-D-glucose linked together by beta-1,4 bonds, and because humans cannot digest cellulose due to the lack of cellulase, it actually acts as a dietary fiber that improves the gut motility. The last one is GAG, glycose aminoglycans, which is a long unbranched polysaccharides made of repeating disaccharide units, consists of amino sugar and uronic acid, usually found in the ECM and the connective tissue. And because they are negatively charged, so they can bind to a large amount of water to form a gel-like matrix, act as a shock absorbents. The GAG can combine with a core protein to form proteoglycan, where it is a major component of the ECM function as shock absorbents and weight bearing. A loss of GAG or proteoglycans can lead to osteoarthritis.
So now I'm going to talk about the function of different carbohydrates. Carbohydrates perform several essential functions in the human body. So the primary function of carbohydrate is to provide energy. Glucose is the main fuel for the brain and the only energy source for the red blood cells, which depend entirely on glycolysis for ATP production. Next, excess glucose is stored as glycogen in the liver and muscles. Liver glycogen helps maintain normal blood glucose levels between meals, while muscle glycogen supplies energy supply during physical activity. This storage system ensures a continuous and reliable energy supplies. Carbohydrates also play an important role in regulating blood glucose levels. Insulin promotes glucose uptake and storage when blood glucose is high, whereas glucagon stimulates glycogen breakdown into glucose when blood glucose is low. This hormonal regulation is essential for metabolic homeostasis. Another key function of carbohydrate is their protein-sparing effect. When adequate carbohydrates are present, the body does not need to break down proteins for energy, allowing proteins to be used for growth, tissue repair, and enzyme synthesis. Carbohydrates also prevent ketosis. In the absence of sufficient carbohydrates, excessive fat breakdown leads to the formation of ketone bodies. So, adequate carbohydrate intake ensures a proper fat metabolism and prevent metabolic acidosis. Finally, carbohydrates have important structural and cellular roles. Cellulose and chitin provide structural supports in plants and arthropods. Glycoprotein and glycolipids on cell membranes are involved in cell recognition, immune response, and blood group determination, such as the ABO blood group antigens. Overall, carbohydrates are essential for energy, metabolism, and cellular function.
My name is Danish, and I will be talking about the digestion of carbohydrate. Firstly, the goal of digestion is to break complex carbohydrates into monosaccharides so that it can be absorbed and used for energy. The site of digestion would be mouth and small intestine, specifically the duodenum. Firstly, the mouth. The enzyme that is used is the salivary amylase, or known as ptyalin. It is secreted by the salivary glands and works best at a neutral pH. So what it does basically, it will break down starch and glycogen to be converted into maltose, maltotriose, and dextrins. But this digestion is short-lived because the food will be swallowed quickly, and digestion will stop in the stomach, where there is no carbohydrate digesting enzyme, and gastric acid will also inactivate the salivary amylase. Next on, it will be digested in the small intestine, specifically the duodenum. The enzyme will be the pancreatic amylase, and it works best in alkaline pH. It will digest starch and glycogen into maltose, maltotriose, and alpha-limidextrins. The final site of digestion is the brush border, which is a layer of microvilli on the apical surface of the enterocytes, mainly found in jejunum. 4 enzymes will digest the product from the digestion in the duodenum, breaking them down into smaller molecules. The 4 enzymes will be maltase, sucrase, lactase, and isomaltase. Maltase will digest maltose into 2-glucose. Sucrase will digest sucrose into glucose and fructose. Lactase will digest lactose into glucose and galactose, while isomaltase will break down alpha-1,6 glycosidic bond on alpha-limit dextrins and further digest it into glucose.
Today, I'm going to briefly explain how carbohydrates are absorbed in the human body. Carbohydrate absorption mainly occur in the small intestine, especially in the duodenum and jejunum, where the intestinal lining has microvilli that increase surface area for absorption. Before carbohydrates can be absorbed, they must first be digested into monosaccharides. Digestion starts in the mouth through salivary amylase, although its effect is short. The main digestion happens in the small intestine, where pancreatic amylase breaks down starch into smaller carbohydrates like maltose and dextrins. Then enzyme on the brush border membrane complete the digestion. For example, maltase converts maltose into glucose, sucrase breaks sucrose into glucose and fructose, and lactase breaks lactose into glucose and galactose. Once monosaccharides are formed, absorption occurs through specific transporters in the intestinal cells. Glucose and galactose are absorbed through a transporter called SGLT1, which brings them into the enterocyte together with sodium. This is known as secondary active transport, and it depends on the sodium gradient maintained by the Na+/K+ ATPase formed on the basolateral side of the cells. On the other hand, fructose is absorbed through GLUT5 by facilitated diffusion, meaning that it requires no energy. After entering the intestinal cell, all three monosaccharides exits into the bloodstream through GLUT2 on the basolateral membrane and then travel via the portal vein to the liver. Clinically, one important example is lactase deficiency, which causes lactose intolerance and leads to symptoms like bloating and diarrhea. Also, oral rehydration salts work well because glucose absorption through SGLT1 helps pull sodium and water back into the body.
Now we move on to the next one, which is I will discuss the key disorders of carbohydrate metabolism. Starting inside the cell with glycolysis, we have hexokinase deficiency that blocks glucose entry, phosphorylkinase 1 deficiency that also called as thyroid disease that slows glycolysis at the red limiting step, pyruvate kinase deficiency that stops the last step, which is reducing ATP, especially in red blood cells. Next is digestion and absorption. We have lactase deficiency that causes lactose intolerance, sucrase isomaltase deficiency that prevents sucrose digestion, SGLT1 defects that causes glucose galactose malabsorption, and GLUT5 defects that lead to fructose malabsorption. For monosaccharide metabolism, we have classic galactosemia that caused by GALT deficiency that leads to toxic galactose 1-phosphate accumulation, galactokinase deficiency that causes cataracts, hereditary fructose intolerance due to aldolase B deficiency causes hypoglycemia and liver toxicity. Next is glycogen storage disorder affect glucose availability. We have von Wieck disease that blocks glucose release. Pompe disease causes lysosomal glycogen buildup. McArdle disease that prevents muscle glycogen breakdown leading to exercise intolerance. In pyruvate and gluconeogenesis disorder, we have pyruvate dehydrogenase deficiency that blocks pyruvate entry to the Krebs cycle that causing lactic acidosis. Fructose 1,6 bisphosphatase deficiency that impairs gluconeogenesis leading to fasting hypoglycemia. Next is systemic regulation disorder. We have type 1 diabetes that prevents glucose uptake and type 2 diabetes that disrupt glucose control through insulin resistance. Finally, the pentose phosphate pathway disorder, we have G6PD deficiency that reduces NADPH that causing hemolysis of red blood cells under oxidative stress. In conclusion, this disorder highlight the intricate regulation of glucose metabolism from digestion and absorption to storage alternative pathway and systemic control. That's all. Thank you.